We are about to go live, so
I'm trying to be multitasking.
So you have any jokes before it starts and you want some audience? That's the moment.
Yeah, we're now live on YouTube.
Yay. Can you see it? Okay, we are about to go live, so you know. Yeah. So you have any jokes before it starts and you want some.
Okay.
Better like this. Okay. Hello, hello. Pendant 5 minutes, je les ai mis tous co-host. And co-host aussi, tes co-hosts.
Are you taking a look to the waiting room, Celine?
Yes, I am. I see just one person waiting here. Yeah.
Can I share my slides real quick just to see if that same problem happens?
Yes, sure. Let's see.
It says I can't screen share actually.
And you are, let me check because I was turning you all one after the other. Let me know if now it works.
Yeah.
Okay. Yeah, sorry, it was on my side.
It's working on my screen.
Yep. Great. And in case Marie-Laure is next to me with all the presentation, so if there is any need of backup, we are here. Okay.
Did that work?
3 minutes.
Go on, I'll just share mine as well, just to be sure. Not that I'm
Yes, it works.
Okay, I'll try mine too.
Just in case, Pam.
Yeah, go. You have 2 minutes, so please, if you want to test anything, just it's the moment.
I lost my PowerPoint. Hold on.
Ok, où tu as son PowerPoint de Pamela avec la keynote presentation.
There it is.
Yeah.
There it is.
Okay. Thank you.
I think, yeah, we are almost ready to go. When you're ready, Anna, the floor is yours.
Well, it's still 1 minute.
Yeah, but just 1 minute left.
Right. Thank you.
Best of luck to all.
Enjoy, everyone.
Thanks.
And thank you all.
Okay, it's already 2. We have already an audience. So I think we need to start. Good afternoon, everyone. For those who may not know me, my name is Ana Casino. I'm the CETAF Executive Director. CETAF stands for the Consortium of Collections-Based Institutions across Europe. And I'm also a Deputy Coordinator of DiSSCo, the research infrastructure that our community is currently building. So it is for me an honor to be here welcoming you all to this meeting, to this public event that aims to present the work that a group of international multidisciplinary experts have been carrying out for the last 3 months. A group of over 60 experts coming from different disciplines, coming from different countries, all linked to natural science collections and volunteering to work, I have to say hard, towards the same goal, trying to identify how natural science collections may contribute to monitor, whenever possible, to mitigate the effects of the COVID-19 pandemic that we are suffering, and most likely to help preventing the ones to come. So, but before getting to the topic, I would like to give you some practical information. We have posted in the chat the agenda of this meeting. As you have been able to look at, we will start with my introductory words, and after that we will have a keynote speaker that will be followed by 4 presentations, the 4 activities in which this task force has been divided. Each presentation will be followed by 5 minutes of questions, in a space open to the audience to place the questions. To make this event as interactive and lively as possible, we have created a Google Doc that is also posted in the chat, in which you may add your comments, your remarks, your questions to either one of the presentations or also in regards to the overall initiative. You may leave your contact and we will try to make our hardest in answering those questions. Certainly, because of the time constraints, we will not be able to answer all of them. So we will try to give answer to the first ones in order as they come in and they are written into in the Google Doc. So don't be shy. We will try to answer as many as possible, perhaps not today, but we will follow up on those and we will tweet, we will use our social media to really come back to you and make comments on your issues and the possibility that we have to connect with you all. So, as I said, don't be shy on the part of the participants. But on the part of the presenters, I have to remind you that you have to stick to the time slots that you have assigned. We have a very, very much constraint in terms of timing. So there will be 1 minute warning announcing that you have to wrap up. You have to have in your final words, and that we will have to finalize with your own presentation. So, well, this is basically my part of the practical information, but let's go back to the topic, the COVID-19 task force. And let's try, I will try to frame it, how and why this task force was created. Looking backwards to earlier this year, by January, even February, most of us, we were already aware that we were facing an unprecedented health crisis, a pandemic that was caused by the SARS-CoV-2 virus, and that goes beyond any geographical or political border, any social status or economic position. We certainly were facing a pandemic that was requiring a collective response from governments and scientists, from citizens and professionals, all working together and on a collaborative way to address this challenge. And the scientific community was there, ready for action. I have to say that the community that is linked to biodiversity data has demonstrated a very impressive impulse and motivation. So COVID-19 and certainly any other upcoming pandemics, because regrettably this will not be the last one that we have to address, is a global issue. And as such, we have to give a global response that may not come other way than from a combination of efforts and resources at international level. Because certainly we have many disciplines that combine their efforts in addressing this challenge. But our natural science collections community has also a say in providing solutions to this. Because it is true that we are not totally certain on the origin, but the scientific community has already strong indications that this is a zoonotic disease. And if this is the case, certainly the distribution, the population ecology, and their interspecies interactions can be of real high value to identify the origin, the propagation, the evolution of the virus, and of course, give support to act accordingly towards this pandemic. Our community in Europe is gathered around CETAF, this unique organization that gathers natural science, natural history museums, botanic gardens, biodiversity centers, academia, that is focused on natural science collections research, and is also at the core of building the DiSSCo research infrastructure. That is a distributed system that will provide access to the priceless information that our collections contain. So with such a supporting group of institutions, we are talking over 130, with more than 5,000 scientists working on collections, that we jointly gather 1.5 billion specimens that represent almost 80% of the biodiversity described worldwide. With such an asset, how could it still be inactive? We couldn't. We recognize that we might not be at the forefront because the biomedical, the health link disciplines need to have to play that role and be at the forefront, but we could still provide an invaluable, a real valuable asset with the data coming from the natural science collections, with the critical and very robust evidence that our collections contain. And this is what we did. We created this task force joining experiences, knowledge, skills, all our data around CETAF and DiSSCo all together with the support of institutions operating in the international arena, like IVIC-Bio, which is our sister initiative in the States, with GBIF, the Global Biodiversity Information Facility, with Spinach, which is the Society for Preserving Natural History Collections, with many others like Platzi and Pensoft from the publishing perspective, that really with GLOBI as well, that is the Global Biotech Interactions Initiative. I think I don't miss any one of our supporters. We decided that we needed to take action, and our community was ready to do so. So this task force was created when we decided to collate and to gather all these efforts. And I have to say that this public event is also, for me, a way to give public recognition to all those scientists that have been working hard and collaborating for the last 3 months and will continue to do so, I expect. Every Friday for more than 1 hour, and during a lot of other meetings in between, they have worked for addressing how the natural science collections could contribute to find solutions in this situation, in this big challenge that we are facing. So though we distributed the task force in around 4 activities, 4 different approaches. One for creating a reference to the animal virus carriers. The second for structuring the way to preserve the viral evidence. The third for building a knowledge hub. The fourth for improving the metadata registering practices. All of them will have their time later on to present their goals, their activities, their expectations, etc. But before getting there, we needed and we wanted today to frame our actions, our activities in a larger picture. Because of course, none of these efforts come from scratch. The natural science collections community has been there for decades, working together, collaborating, making impact and having raising awareness in the public society and in other disciplines of the high value of the natural science collections, of the information that we contain and that we put there for the benefit of science and society. And therefore, to frame, to present that landscape, to really give value to the natural science collections and how they contribute to biomedical sciences, in this case for addressing this pandemic, we requested the participation of a keynote speaker. And I have to say that we are honored, and for me it's a personal pleasure to have with us Pamela Soltis. Pam is is not only our keynote speaker of today, she has been collaborating with the task force since it was started. Pam is a distinguished professor and curator of genetic resources and molecular systematics in the Florida Museum of Natural History at the University of Florida. And she serves also, among others, as director of research for IVIC-Bio in the States. Her main research interests are, among others, the use of digitized natural history collections in biodiversity research. Her work has addressed novel uses of herbarium specimens, for instance, including niche evolution and phenological analysis using machine learning systems and images of the herbarium specimens. A long list of works that include even some outreach products linking arts and science, so a very multidisciplinary approach to natural science collections. She served as president of the Botanical Society of America and also of the Society of Systematic Biologists back in the recent past. And she is currently president of the American Society of Plant Taxonomists and of the International Society for Phylogenetic Nomenclature. She's a member of the US National Academy of Sciences and a fellow of the American Academy of Arts and Sciences. I had to read it, Pam. So it is really my pleasure to introduce and to give the floor to Pam. Welcome, Pam, and thanks for being today with us. The floor is yours.
All right. Well, thank you, Anna, for the very kind introduction. And thank you also for the invitation and the opportunity to be able to speak today. I'd like to begin my presentation by also, let me just get my laser pointer on here with the right color. Green, of course, because I'm a botanist. There we go. I'd like to begin really by thanking Anna, Dimitris, and Wouter again for organizing the task force and for casting such a broad net, one that went clear across the Atlantic to include some of us from the US and also from Brazil. Now, the immediate motivation, as Anna mentioned, for the task force really was the current pandemic situation. And what this raised for many of us, though, was the thought about how can the natural history community, or I guess as you refer to it more in Europe, the natural science community, contribute to better understanding of the pandemic and perhaps contribute to solutions. So some of the questions that our community can help focus on include, what is the natural host of SARS-CoV-2? Where did SARS-CoV-2 originate? What is the pattern of transmission? And in doing so, we can look to our natural history or natural science collections to provide us with baseline information that could be useful. In addition, many of our collections also have associated with them genetic resources. And then there, of course, is a vast literature on all sorts of aspects of natural history and natural science. I'd like to spend just a couple of minutes considering genetic resources repositories. Now, as I mentioned, many of our museum collections also have such genetic resources, and these collections may provide a range of materials from tissues to DNA to RNA, and they can provide sources of pathogens as well as the pathogen DNA, as well as, of course, the host DNAs as well. Now, in 2014, IDIC-Bio developed a directory of genetic resource repositories in the US. And as we began to think about what some of the resources could be for addressing the pandemic from the natural science perspective, we realized that it could be useful to update this directory. So one of our graduate students, Maria Cortez, worked with me to update the directory. And we worked particularly with the mammal community, John Dunham in particular, to revisit possible genetic resources repositories in the US. And our list went from 54 to 78 institutions, each represented by the name and description of the collection, as well as contact information. And again, really the goal with this effort was to identify additional resources that might be useful for any sorts of studies involving the natural hosts of and possible distributions of the SARS-CoV-2 pathogen as well. And we wanted to make this information as discoverable as possible. So if we return to some of these questions that were motivating our activities, the big question then, as listed at the bottom, is, well, how do we actually mobilize our data and information? And this leads, I think, to thinking about some additional, more specific questions, such as, where are the bat specimens located that correspond to some of the possible hosts? Which museums house those specimens? Where do these bats occur in nature? And what about the synonymy of the bat species? And can we put locations on a map to learn more about their habitat? And in so doing, perhaps also learn more about possible interactions that these bats may have with their local environments. And then where do I find literature on all of these topics, and how do I incorporate that literature knowledge? In addition, are there genetic resources that could be available for these bat species, from which the viruses or their DNA might be extracted and identified? And what sequences are actually available? And what metadata are attached to those sequences? Now, although we may think that we have the necessary information and that this information is available in various formats, in fact, what we find is that there are a lot of gaps. In fact, when we try to investigate really any of these separate components, we find that each of these is a very difficult step. And this brings me to a few comments on our task force and the activities. And I'd like to use this again to help frame how we can identify ways to move forward, both on this single topic of COVID-19, but also what the implications are then for the larger biodiversity community. Now, when we started the task force back in April, we suspected that there were some gaps in data availability or utility, but we weren't really sure what they were. We had an idea that most of the information that we thought we might need would be out there, but we weren't exactly sure how to access the information and how to bring it together. So as Anna mentioned before, there were these 4 activities that took shape. And what I'd like to do now is just mention briefly, again, the goals of each of these separate activities, and then each activity group will provide an update on its activities shortly. So activity 1 was focused on identifying priority questions that biodiversity scientists should explore about pathogen origins, spillover possibilities, and ecological correlates. Activity 2 had a goal of developing guidelines for the preservation of microorganismal samples and host specimens in natural history collections. And as part of these guidelines, there were topics of like the types of samples to be preserved, optimal storage methods, and the types of metadata that would be most valuable. In addition, they were interested in tailoring their efforts to accommodate different audiences, from field biologists and curators to biomedical scientists, and to highlight the importance of vouchers as supporting references for future studies. And we'll come back to the topic of vouchers shortly. Activity 3 was focused on developing a knowledge hub on COVID-19 and specifically on liberating information about mammal host and virus associations through digitizing, extracting, and indexing known biodiversity data. And activity 4 focused on metadata requirements, including defining and refining the metadata requirements that should accompany viral and host information, along with identifying the guidance and tools required to capture these metadata in GenBank and affiliated databases, and then also the actions required to socialize across the scientific community. So again, returning to this question of what information do we need, we really need to think a little bit more specifically about some of these issues of host specimen availability, geographic distribution, taxonomy, and so on. And if we could pull this information together, we could provide important insight into the possible origin of the pathogen, perhaps the modes of transmission, patterns of transmission, and so on early on in the disease, and thereby help mitigate further novel spillover to human. to humans. And we also think that this could help identify possible future disease outbreaks before they reach pandemic status, and also to help us understand disease outbreaks more fully. Of course, these sorts of activities require collaborations not only among different sorts of natural history and natural science collaborators, but also collaborations with virologists, pathobiologists, and people from the biomedical community. Now, this argument for greater roles of these natural history or natural science collections in biomedicine, particularly with regard to these particular pandemic issues, have been made in a number of recent publications. So just a few weeks ago, 2 papers appeared in the journal Bioscience. One was an editorial led by Jennifer Zaspel and with a number of colleagues. And the second one, published as a viewpoint in Bioscience, they appeared in the same issue, was developed by members of this task force and some additional collaborators. Now, this viewpoint article made a number of recommendations, such as best practices, the need for developing best practices for sample preparation, including voucher specimens. We also recommended improvement in metadata requirements, particularly in the use of persistent identifiers. We also recommended improvements in infrastructure, both physical and cyber, in support of both physical and digital objects in terms of data and specimens. And then we also recommended the need for open science and collaboration among communities. And finally, the strong recommendation for financial support from a range of partners, including various government agencies and private foundations and other sources of support as well. Now, in addition, The Conversation, which is a lay publication, picked up an article that refers back to our viewpoint article and has helped to raise the awareness of the importance of museums in addressing pandemics. And this article has received close to 11,000 reads in just the last couple of weeks. So I would like to emphasize at this point that the COVID-19 pandemic is just one societal problem that is related to biodiversity. As the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services reported last year, nature and its vital contributions to people, including biodiversity and ecosystem functions, are deteriorating worldwide. And in fact, they identified the direct and indirect drivers of change that have accelerated during the past 50 years. And they note changes in land and sea use, direct exploitation of organisms, climate change, pollution, and invasion of alien species. Now, in fact, oops, that went too far. In fact, biodiversity is at the core of a number of important societal problems facing us during the 21st century. And one of the most fundamental, of course, is the loss of biodiversity. But associated with the loss of biodiversity come a number of other societal problems associated with this. And one of these has to do with loss of ecosystem services, or the benefits provided by ecosystems to humans. And just a few obvious examples here include the maintenance of clean water, flood resistance, the pollinators for our crops, sources of new foods and medicines, beauty, and also many more ecosystem services that we all benefit from when we have healthy ecosystems. But in addition to the loss of biodiversity and the attendant loss of ecosystem services, are numerous and unpredictable at this point responses of species to climate change. We also have increased opportunities for invasive species, and we see this all the time. This is a huge economic as well as ecological problem. Emerging pathogens, of course, we're witnessing that at this very moment. But there are also other environmentally induced health issues and of course, threats to food security. All of these relate back to biodiversity, and biodiversity science and our collections therefore have important roles to play in addressing these questions. So the solution, in part, lies in robust interconnected infrastructure linking data across repositories. And I've just listed a few here. We have museums in the center there, but the digital versions of those specimens as found in GBIF and IDIG-Bio and other repositories. There are textual data in the Biodiversity Heritage Library in Zenodo, all sorts of data sets in Dryad, environmental information in WorldClim, traits in the Open Traits Network, as well as many other repositories, and genetic information in NCBI and its partner associations. So in order to make this happen, though, I suggest that there's a need for innovations, several innovations, and their implementation. And I'd like to spend just the last few minutes breaking down what is represented here in this image. So first of all, we have the extended specimen as described by Michael Webster and further described by the Biodiversity Collections Network in a publication that came out last year. We also can think of this as the holistic specimen. And the premise here is that we need to really develop and implement this concept more fully. And that involves linking the underlying museum specimen with all of its derivative parts and data. And this could include DNA sequences of the specimen itself, DNA of any pathogens, other information on pathogens, scans, images, various sorts of information, isotopes, chemistry that might be available in other databases. But to make those connections effectively, we actually need to have identifiers for all of those that will point back toward the voucher specimen, the museum specimen that is the nexus for all of this information. And of course, identifying the appropriate method for assigning persistent identifiers is also a challenge. And this shows one example as proposed in a paper a few years ago that includes, among other people, Anna and Quentin, and I'm not sure who else from our task force may be authors there, but this represents one possible way forward. But there's a long way to go before the community is ready to adopt this or any other sort of opportunity for identifiers. We're also actually very short on data information for a lot of specimens and species. And so
Hello,
I think,
sorry to interrupt you, just letting you know that your time is almost up.
okay, thank you. I'm just about there. The use of artificial intelligence and machine learning to develop new trait sets. And I'll just point you to a special issue of Applications in Plant Sciences, that is focusing on this very topic this month and last month. We also need open data. And in this case, open data need to be available and open, but also fair, findable, accessible, interoperable, and reusable. And finally, we need the tools and cyber infrastructure that will allow us to integrate and analyze heterogeneous data and provide a robust backbone to support these analyses. So to conclude, our natural history collections and associated data are important resources for the study of COVID-19. They also, in conjunction with biodiversity science, can help predict and possibly prevent future pandemics. And the lessons that we're learning from this recent experience of trying to integrate data can be extended to additional issues, biodiversity related issues for the coming century, as many of our societal problems do relate to biodiversity. So I recommend better data integration and cyber infrastructure, and that will require ongoing innovation and implementation of new approaches. And I'd like to just end by thanking all of the task force members for inspiration, creativity, and dedication. It's been a real honor working with all of you, and I look forward to continued efforts going forward. Thank you very much.
Thank you, Pam, so much for this presentation and for allocating natural science collections at the center of many societal challenges, and for addressing those, for pointing out the need to have a very robust infrastructure that enables to have the data allocated and openly accessible. So, well, this is now the time for questions, perhaps to your presentation, Pam. Debbie, do we have any that we can address to her?
We do have one, and I think it addresses the broader topic of what's at the root of these host pathogen spillover events. Because the question, Pam, is how can natural history collections underpin actions on forest and the ecosystem landscape restoration? So getting at the sort of root of how can collections help in events?
So there are a lot of, actually a lot of topics in that question.
Yep.
I think maybe I'll start just from the perspective that our collections are, our communal collections, our global collections are global in scope and distribution. And this allows us to actually, once everything is connected, to trace pathways of movement and transmission, regardless of the organism, particularly if we have some sort of genetic marker. But it also gives us an opportunity to look at, from some perspectives, at the density and distributions of individual species. And so I think we actually are probably underusing our collections from an ecological perspective. So I think there's a lot that we could learn about basic ecology by more advantageous use of our collections. And I think that lies maybe at the center of the set of questions that I think were included in that. I may not have actually touched on all parts, so I'd be happy to follow up a little bit more if necessary.
There's one follow-up question Brad put in the chat, and I think he's looking for specifics, I'm guessing here, Brad. I'm putting words in your mouth. What role do other collections like Herbaria have to play in this effort? So how can Herbaria help in this particular effort?
Right. So it may seem a little odd, Brad, that a botanist has been giving this talk, but in fact there are several botanists on the task force, and I think it's our commitment to collections in general, and also our interest in data integration that has allowed us to be able to make contributions to this effort. Of course, the real stars of the show are the virologists and the mammalogists, and we'll hear more from some of them coming up. But with regard to understanding possibly the natural distributions of microbes that are non-pathogenic in their hosts, but become pathogenic in humans, it's important also to understand their environments. And so the plant specimens from the same regions help to define what those habitats and ecosystems are, and could also be important in predicting additional sites of outbreak where ecosystems have become damaged or fall apart. So I do think that there are roles for eventually understanding how all species in a community are interacting. And I think we'll hear more about that a little bit from Nate later on.
Right, we will have the opportunity to hear about that when we talk about the third activity. Now is the turn for the groups. Thanks a lot, Pam, for such an incredible presentation, for sharing your ideas, your insights, and certainly to help us to strengthening the connections between the different parts of the world, because certainly our collections, as you said, do not understand of frontiers or borders, but they stay there all together. Thanks a lot, Pamela.
Yeah. Thank you.
As I said, now the time for our activity presenters. The first one, the first activity, as Pamela was presenting in her presentation, deals with animal virus carriers. The group has circulated a questionnaire through a lot of networks, addressing a very, very large community of scientists to identify priority questions that biodiversity scientists should explore about pathogen origins, spillover processes, and their ecological correlates. Professor Danilo Russo will present the work of the group. Danilo is an associate professor at the Naples University Federico II in Italy, and is also an honorary member at the University of Bristol in the UK. His interests include a lot, from habitat selection, resource partitioning, sensory ecology, social behavior, evolutionary biology, biogeography, and invasion ecology, among others, I may say. Most of his research focuses on bats, but he's also working on a range of other model organisms. Since 2019, he has been chairing the scientific committee of the UNEP Eurobats Agreement. He's an active publisher, he is an editor-in-chief of the zoological journal Mammal Review, and he has conducted field work in many regions and environments across the globe. Danilo, the floor is yours for presenting the activity 1 results. Thanks a lot.
So thank you very much indeed, Anna, for the nice presentation and for the chance to speak today and present our work. The work we've been doing is a survey of research priorities on pathogen spillover and the ecological correlates of these processes. I'm presenting on behalf of my colleagues, Sandro Bertolino, Severin Tis, and Martin Van Hoof. We've been working quite a lot together over the last few weeks. And I will start from a message we've been bombed with during lockdown, that the word crisis in Japanese is made of 2 kanjis, one meaning danger and the other opportunity. Well, in fact, this is true because crises should be seen also as sources of opportunities. And COVID-19 pandemic was a big tragedy, of course, worldwide, but also a chance to learn something about prevention of future pandemic. Because, of course, we'll deal with other pandemics for sure. That's not the likelihood. It's certain that this will happen sooner or later. So we should be ready for that. And within this context, research is key to address questions that will inform prevention strategies. So our main objective was to identify priority questions that biodiversity scientists should explore about pathogen origins, spillover processes, and their ecological correlates. To do so, as Anna was anticipating, we sent a questionnaire to reach out as many experts as possible worldwide. And we've been still collecting material and starting to analyze it. So I'll present our preliminary results today. Where is the link with natural history collections? Well, the link is that being natural history collections biodiversity infrastructures, actually they may act as fundamental sources of answers to respond to future pandemic events that the ever-growing human population will inevitably face. So what we are going to do as an ultimate goal of our activity is to search links between the priority questions we are collecting and the potential of natural history collections. As I said, we're a small bunch of people, a core team of 4 people, including medical anthropologist, Severin, an evolutionary parasitologist, Martin, an invasion ecologist, Sandro, and myself, a bat ecologist. But the group is open to collaboration for data analysis. So we are fully open to people who are willing to share their expertise, especially if someone may help with data analysis. That would be really great. So please contact us if you want to join us and get aboard. Of course, the very strength of our work is the interdisciplinary expertise of respondents. This is the cover letter and very beginning of the questionnaire we circulated over the last few months. The questionnaire is about priority research questions on the ecology of pathogen spillover events. And so far we got something like 100 respondents, but further input is still highly welcome, especially from less represented expertise such as policymakers, social sciences, biomedicine researchers, veterinary medicine researchers. And in terms of geographic scope, it would be great to get more answers from the southern hemisphere, because most of the respondents were from northern latitude. It would be great to have a more global coverage if possible. We've been working on the analysis of the answer to date. And as for the challenges we had to deal with, I would say, first of all, the survey fatigue, the COVID-19 fatigue. We've been snowed under with questionnaires during lockdown, and it's probably been a bit too stressful for many of us to respond to all questionnaires. So we got a little bit of resistance, especially in the very beginning of our questionnaire circulation. And there's also a bias in response rate that I'll show you in detail in a second, which is probably due to our connection to certain networks. So although we covered a diverse range of experts, as you can see, most experts are ecologists or zoologists, then virologists, and then other categories were still present, but way less represented. And also in terms of geographic scope, we got a bias towards Europe and North America, again, probably reflecting the nature of our networks. Although we got some responses from Africa, but very few from Asia and South Central America. So it would be great, again, to collect more respondents from those regions. As for data analysis, we've been fortunate to have Severine aboard, because she is an expert of using software which works specifically on this kind of data, and Vivo in particular. It's software meant to analyze non-numerical data, which in this case is scientific questions. And it allows to classify and sort data, examine relationships and trends in the data. So we used a thematic approach. So we identified major topics through coding by researchers involved in the study, ourselves following an inductive approach. And here you can see what we did. Based on a first subset of data, 459 questions and 80 respondents, we got a classification into main topics. Here you can see that the spillover processes and mechanisms were the most represented category. And some questions were coded in more than one topic or subtopic. So some questions could actually be labeled according to different topics. So they fell into 2 or more categories when needed. And we also had a category of non-relevant questions. Whenever a question was not clear or not directly related to the survey topic, etc., we classified it as non-relevant. But we'll get back to those questions in the refinement process to see what we can do with it. Here's a list of subtopics related to spillover processes and mechanisms. So what we got was actually within each main topic, subtopics, and so on, we had to refine our classification, because, for example, among the global changes related questions, we had questions that were more specifically referred to land use change, for example, wildlife consumption and trade, climate change, invasive species, and so on. So we got this further categorization within main categories. What next steps are? Well, first of all, we have to start data analysis. We're still collecting material, by the way, so the survey is still open, but we are starting to explore methods of data analysis, and we'll come up with an article preparation, I hope quite soon. But in long term, the goal of our activity is actually to prioritize how to investigate recent and collection held biodiversity in the context of zoonotic prevention and response, to achieve a better integration of biodiversity science and ecology into biomedical research and health policy. We also actively search cross-links with the other working groups and with the One Health EcoHealth scene, which is really very important nowadays. So, as I said, data analysis is ongoing, but the first results show that the scientific community has great concern about the ecological significance of COVID-19 pandemic and similar events. So that's something which has been sort of overlooked by the media, but it's very clear for many of the scientists who responded to our questionnaire that the very problem, the very cause of pandemic events such as COVID-19 lies actually in environmental issues. And this is why our exercise is actually also building up a plea from the scientific community for investigating the environmental correlates of pandemics and develop multifaceted interdisciplinary prevention and management strategies. I would conclude really saying a big thank you to the CETAF-DiSSCo initiative, because they offered us a unique chance to join forces, meet new colleagues and friends, I would say, and develop fantastic collaborations. So I'm pretty sure that many future collaboration will stem out of this excellent initiative. Here you have our email addresses in case you want to discuss with us what we've been doing, or you want to think about joining us, especially if you are an expert of dealing with this kind of data sets. You're more than welcome to contact us and we'll discuss the matter. Thank you very much for your attention.
Thanks a lot, Danilo. First, to stick to the timing. Secondly, for appreciating how important is collaboration across disciplines and also across countries to come up with sound knowledge base that could support to address this pandemic, but also future ones. So certainly, this is a very, very nice key message to take back home. Any questions to Danilo, maybe?
No direct questions at the moment, but I do have one quick one. I think we have time for, I hope. Is the survey still open, Danilo?
Yes, it is. Yes, it is. We are still collecting questions, and we hope to reduce the bias in terms of geographic scope and expertise as well. So yes, definitely yes.
So could you put the link in the chat to people?
Yeah, sure. We will. May I ask Sandro? Probably Sandro has it handy. Sandro, do you have the?
Yes, I'm here. I will add the link to the chat.
Thank you, Sandro. And maybe some people here can help you distribute it as well to their colleagues.
That would be fantastic. Thanks.
Yeah, good point. Well, I would like to take this opportunity of having 1 minute left, because as you can see, the work has not stopped. It has not been finalized by today. It is true that the COVID-19 task force had a framework of 3 months to come up with tangible results and to really produce actions. It is true that we want to continue with this work. It's something that has been an opportunity for many of us to find people with whom we can collaborate cross-disciplinary. So our intention is to keep continuing and keep working together. First, because some additional products will result from our work. And secondly, because there are some grants, some projects that have been like in spin-offs of this task force that will ensure the continuity of our work, both in Europe and in the States. In Europe, under Synthesis Plus, we will have most likely the proposal approved to digitize and add metadata to certain groups of bats held in collections across Europe. And in the States, thanks to several grants from the National Science Foundation, there will be projects running for more months, for the months to come, in terms of adding georeference and some other metadata to the existing databases on bats. So as you see, the works will continue and our activities will be still there. with their doors open to welcome new contributors and new collaborators. So please keep tuned with those. If any question that could be added to the Google Doc, Deb, or shall we move to the second activity?
I believe it's time to move on.
Okay, so the second activity, the second approach to the impact, the value of natural science collections when addressing this COVID-19 pandemic deals with what we have called the preservation of the viral evidence. It has to do with developing guidelines for linking host and microorganism parasite samples and data. Kendra Phelps will present the work of this group. Kendra, she's a research scientist at the EcoHealth Alliance and works also as a biostatistics consultant at the Texas Integrated Diving and Ecological Studies Laboratory in the States. She has over 18 years of experience conducting field-based research on the ecology and health of wildlife with a focus on bats. In fact, interactions between humans, environment, and wildlife are at the core of her research, focusing, among others, in identifying specific environmental and ecological drivers that enhance pathogen persistence and transmission between bat hosts. So, Kendra, the floor is yours to present the activity of the second group. Thanks.
There we go. Well, thank you for that introduction and good morning, everyone from New York City. And as Anna mentioned, my name is Kendra Phelps and I've been working with EcoHealth Alliance for several years now. But my goal today is to provide you with an overview and an update of Activity Group 2. And as our group name is Preservation of Viral Evidence, I want to point out that this doesn't quite really encompass all of the activities that we've been involved with since the creation of this group in response to the COVID-19 pandemic. Initially, the main objective of our group was to develop best practices for preserving viral samples in natural history collections. And this targeted approach to viruses was driven largely by the realization that a coronavirus was the causative agent for the COVID-19 pandemic. But over time, our focus broadened in scope to include other microorganismal samples, such as bacterial samples and ectoparasites and helminths, but also to highlight the importance of vouchering host specimens in conjunction with these microorganismal samples. And specifically, we aim to identify what are the optimal types of samples that should be preserved and in what storage methods. And also not to forget to collect the critical metadata that should be included with the samples, all while considering how these guidelines should be tailored to accommodate the diverse audience needs. But another equally important objective of our group is also to emphasize the importance of linking researchers who conduct host pathogen research with natural history collections, so that we can develop a more holistic approach to characterizing, mitigating, and hopefully preventing future pandemics. So who is in Activity Group 2? Well, we're quite a rowdy bunch of researchers that specialize in a wide diversity of study topics. So from evolution and ecology, taxonomy and systematics, to even food security and museum informatics. And while many of us have a strong affinity for mammals, there are other members in the group that study things such as parasites, birds, or even fish. But many group members are also associated with natural history collections from around the world, including Sweden, Brazil, Finland, and then also multiple collections here in the United States. So to achieve our objective, the first aim was to develop an online survey to gauge the current vouchering practices that are used among microbiologists. And by microbiologists, this is a broad term that I use to include parasitologists, virologists, and so on. And we were also curious, what were their previous experiences with collaborating with natural history collections if they have. So for roughly 2 weeks, we've been administering an online survey via social media. And we've gotten a decent number of responses, but we're also waiting for approval from the American Society of Microbiologists so that we can distribute this survey to its hundreds of members. But I want to share with you some of our preliminary findings. So as of Monday, 112 individuals from 25 countries had completed the survey. And when asked about their field of research, we were a bit surprised to find out that over half of the respondents combined studied systematics and evolutionary biology, as well as ecology, while only about 16% of respondents study virology and microbiology, which is more of our target audience. So what type of microorganisms are the respondents studying? Well, nearly a third of respondents indicated that they study helminths. So these are parasitic worms and flagellates. And the second most popular study organism were bacteria. Interestingly, though, a comparative low percentage of respondents actually are studying viruses. This is particularly interesting considering the current pandemic is caused by a virus, a coronavirus, SARS-CoV-2. Respondents also indicated that they do collect these microorganismal or biological samples, and they're doing it in 97 different countries around the world. But where do these samples end up? Well, nearly 2/3 are permanently archived, but only about half of those end up in natural history collections where they're accessible to a broader scientific community. But we also wondered, why doesn't the other third of respondents archive their samples? And it turns out that it's largely due to a lack of institutional infrastructure that is the major driver that's limiting respondents from being able to archive their biological samples. Now, if we turn to vouchering practices of host species, we're a bit surprised that actually the high percentage of respondents that included lethal sampling of host specimens as part of their study. So this is both targeted sampling as well as opportunistic sampling. Similar to microorganismal samples, we're interested to know what percentage of our respondents are archiving these host species. In a bit of reversal, fewer respondents were archiving the host species in comparison to microorganismal samples. And for those that did archive host specimens, where were they deposited? Well, again, over half are deposited in natural history collections, so accessible to other researchers, but there remains half of those host specimens that are housed in collections that are not accessible to the broader scientific community. And similarly, those that did not voucher hosts were again limited by institutional infrastructure largely. And this probably contributed to a lack of tradition for vouchering host specimens. So it's kind of a circular issue. Lastly, we asked respondents if they collaborate with natural history collections. And surprisingly, there was an equal split of those that do and those that don't. But what I find interesting and the greatest prospect is that the 10% of respondents that we're not sure how to collaborate with natural history collections. And here's one person's statement that both highlights that there is a willingness to collaborate with natural history collections, but that we need to provide them with resources and how to do that. So this brings us to our second aim, which is to draft a manuscript that outlines for host pathogen researchers what are the gold standards for archiving specimens to be deposited in natural history collections. And by doing so, this would facilitate repeatability and extension of future research to prepare for what could potentially be another pandemic. So in the manuscript, our main messages are first to highlight the importance and the best practices for vouchering specimens. And this applies to both microorganismal samples as well as host species. And to do this to archive them in natural history collections. And we'll use examples of collaborations, prior collaborations with natural history collections that have led to the discovery of host pathogen associations using sometimes decade-old specimens. And then we also want to provide some resources, such as a detailed listing of current museum holdings that are ripe for future pathobiology studies. So some key messages that I want to leave you with today are that we need to facilitate collaborations among stakeholders. So this includes those that study microorganisms, such as virologists, parasitologists, those who study the hosts, such as ecologists and systematists, but also with natural history collections. And this is all in an effort to facilitate and inform zoonotic disease risk assessments. And if you look at the specimen in the center of the picture, this archived specimen forms the core, and this is an example of how archived specimens form the core of a large multidisciplinary source of information that can be used to address questions beyond those related to just host pathogen dynamics, but also a slew of other equally important questions that are detailed very well in this figure. So how do we work together to achieve robust science and pathobiology that better prepares us to deal with the next pandemic? And there are two obvious answers. One is to encourage researchers to archive both microorganismal samples and
Kendra, I think.
Almost done, and host species in natural history collections. So this, again, enhances the reproducibility and extendability of their research, particularly in light of taxonomic changes and evolving technologies. And the second solution would be to encourage natural history collections to maximize the diversity of sample types that they're curating. But ultimately, this would require bolstering the current infrastructure that is in place, but also building capacity in regions that are lacking infrastructure, which was highlighted by many of the respondents as the reason behind why they aren't able to voucher their specimens. And I'd like to end on that note and put in a shameless plug for our survey if you're still interested in participating. And I'm open to questions. Thank you.
Yeah, sorry for the bell, Kendra, we couldn't find another way. So time was up. Thank you very much for your nice presentation.
Thanks.
Hi. So
certainly. Debbie, we
do have two questions. Two questions. Great. So Kendra, one of our participants asked if you have reached out to Jennifer Zaspel and Katja Seltman of the Parasite Tracker projects to coordinate best practices relating to capturing host associations in natural history collections. And I would help you out there, Kendra. Do you know these people? Do you? No. Okay.
Sorry, I do
not.
But
do you know Jorrit Polen from our task group here? Yes. And you know Nate, and okay, so they're connected. Globy is that Jorrit has built, our global biotic interactions, participates in with the people that we're talking about in this questionnaire. So maybe they could help. The answer is yes. I can help you. They are connected because Jorrit's in this working group and also works with them. And so we can follow up with you and on that question afterwards, help you out there. The next question for you, it says, what is the stance and activity of EcoHealth Alliance with respect to vouchering specimens?
Well, I can speak to my personal experiences at EcoHealth Alliance. I conduct a large disease surveillance project focusing on bats in Western Asia. And as part of that work, we don't do targeted host sampling, but we do voucher opportunistically. And we pull tissues, we do full necropsies and pull tissues. And then we also preserve the remainder as an alcohol prep specimen. And ultimately, those specimens are destined for the American Museum of Natural History. And that's largely due to the lack of infrastructure in the countries that the 7 different countries that I work in Western Asia. So while we encourage non-lethal sampling, because we do process thousands of bats on an annual basis, we do archive host vouchers when possible and try to pull fresh tissues that are flash frozen in the field. So we try to make the best of what we have and use every part of an individual.
Do we have time for one more question?
Yeah.
Talking to the last.
Quick one.
Thank you. Yeah, thank you, Kendra. The last question here is, are you also interested in principle in plant hosts for microbial pathogens?
Sure. I mean, that's not, honestly, that hasn't been on our radar because, as I mentioned, we work on mammals mostly and vertebrates largely. But obviously that's an important aspect because plant pathogens can also have both consequences for human health, but also economic consequences as well. So they are important as well. I don't know how big of a chunk we want to try to tackle in our group, but it's definitely something to consider.
Thank you.
Great. Thanks, Kendra. That was really amazing to see that there is a need to facilitate infrastructure, to allocate resources that are not still connected to natural science collections in the museum. So we have to look forward and to work hard in that respect. Thanks, Kendra. So now is the time for the next activity group. Number 3, they have been working on a comprehensive knowledge hub. By digitizing, extracting, and indexing published data, they are aiming to liberate thousands of mammal host virus associations. So this is the time for Nathan Upham to present the work of this group. Nathan is an assistant research professor at the School of Life Sciences at the Arizona State University. He's also associate curator of mammal collections at the School of Life Sciences, and is also a research associate both at the Department of Ecology and Evolutionary Biology in Yale and the Integrative Research Center at the Field Museum of Natural History. His study is centered in evolution, ecology, and biodiversity from a spatial and temporal perspective by integrating data from DNA, fossils, and species traits to investigate when and where groups of species originated, at what evolutionary rates, and in relation to which paleo-environmental factors they are connected. So, Nathan, the floor is yours for presenting the results of the group activity 3.
Thanks, Anna. Yeah, very nice. Really great to hear from all of you this morning or afternoon as it is in Europe. Okay. Great. So yeah, so thanks again. And I think the title of our task force is this Biodiversity Knowledge Hub, and it's been kind of the other way that we've been thinking about it is liberating biodiversity data from the lockdown of COVID-19. Just want to acknowledge our many contributors to this task force. Has been definitely a group effort, and I'll talk about different contributions throughout this presentation. But I want to kind of start with, framing this as, obviously we have this coronavirus, novel coronavirus, SARS-CoV-2, definitely a mammal host. We know that. We think it's a bat host, most likely Rhinolophus, these horseshoe bats, trying to pinpoint exactly which species it is. And then there's these kind of major research questions that emerge. And in particular, our group was focusing on this, what is the risk of future spillover events, specifically of zoonotic viruses from wild mammals into humans, and which mammal species in particular. And so we have this kind of event, we formed the group in April. We've got Nancy Simmons, a world expert on bats in our group. And we realize that she's locked out of her office, right? She can't access all these rare books that are in the American Museum of Natural History. And how do we find this information? And it really it caught us unprepared in a way that is quite interesting and kind of speaks to the broader issues in biodiversity data. It's a lot of it is very locked up, a lot of it is in old books, and a lot of it is not interconnected. And so we started kind of thinking about this more broadly, right? So we have observations of organisms that are either vouchered in museums or non-vouchered, but maybe with photo vouchers or with expert IDs and different types of databases. But really the biodiversity literature is a key to understanding the ecology of the organisms and really biodiversity knowledge in general. And so the fact that only a portion of this literature is digitized means that we only have a portion of the biodiversity knowledge digitally available. So humans have discovered lots of things, but we can't analyze them in any kind of interconnected way on computers until we really go down this path fully. And so the goal of our group has been to digitize, extract, and index this known biodiversity data, specifically as relates to viruses and mammal hosts, and kind of focusing on taxonomic treatments and museum specimens in particular. And so this kind of interesting loop between thinking about the mammal hosts, the species names, and also old names for those species, the synonyms, digitizing taxonomic treatments with in Platzi and Zenodo, also pulling treatments out of Pensoft, which is a publishing software. I'll talk more about each of these. And then kind of working to, working to connect so using those names as sort of keys to search the biodiversity literature and in databases, and then connecting those biotic interaction informations in GLOBI. So GLOBI is this global biotic interactions database that is really pulling from lots of different resources, and we'll be talking about that more as well. And so just to kind of step through, this is sort of the technical slide with Platzi, but important to realize that what's happening here is digitizing kind of analog literature and then also extracting it. And so the way this is happening is interfacing with Zenodo, which is this kind of unlimited data repository, and there's a specific portion of it called the Biodiversity Literature Repository, where we can. So this is the work led by Donat Agosti to really extract information from these articles, give everything a DOI, link it up, upload it to GBIF, and then that can all be pulled into GLOBI and available for research questions. And the kind of other side of this, right? So Platzi is focusing on old articles in this kind of legacy literature, and then new publishing is happening in a really exciting way with Pensoft. So this is being called semantic publishing, basically the idea that you have a structured data that is within the article, but that can be easily extracted by different types of web services and connected. And this really is the way forward, right? We can't rely on experts just reading a giant corpus of literature and then knowing things in their head. We need our computers to be able to do this work. And so this is some of the outcomes of this partnership. We have a new custom metadata on Zenodo that allows for more intricate kind of intricate terms, and specifically with host virus interactions. There's this Kobiho community on Zenodo that has been kind of manually curating interactions. And then Platzi has done an amazing effort digitizing entire mammal catalogs, including these old books. First mammal species of the world and before mammal species of the world. And then the entire Handbook of the Mammals of the World, Volume 9, especially the Rhinolophidae chapter. All these treatments are now on Platzi, in Zenodo, linked up on GBIF, and then the data has been extracted into Globi. Similarly with Pensoft, those interactions are flowing now directly into Globi. And once the links are now established, and we can actually, they'll continue to be updated. I just wanted to show that this is a portion of what Platzi is doing. They're actually extracting all these images and indexing them. So these all have individual DOIs, and the books kind of come alive in this process. So this is what the data looks like into Globi, not only from Platzi and Pensoft, but also these newly indexed databases called BATbase. There's this DROD-Vir and DBAT-Vir that are curated out of China, and CBI virus, obviously, and then some existing data sets. And so as a result of this, we put together this data publication that's now on Zenodo. You can go and check it out. I'll show you kind of a summary. 85,000 total interactions have been liberated in this process. And if we kind of parse this to the specific host virus interactions, we get 4,000 unique name combinations after synonymies of host virus name combinations, which comes out to these numbers of mammals and viruses, which by comparison, so this 2017 paper out of EcoHealth Alliance, we've increased the number of mammals by over 100 and over 1,000 unique virus names. And admittedly, some of those viruses are going to be redundant, but this is definitely a path forward. And so kind of the next steps that we're looking at is aligning this information with the mammal tree, this new phylogenetic hypothesis that I was involved in putting together, and really looking at quantifying the risk of viral spillover. And so kind of key lessons from our group, there's been two of them in particular. And one is that we can liberate this old taxonomic data out of old literature, but it's very expensive, it's technically complex, and we really don't have the resources to continue doing this for the legacy literature that's growing, because things are getting published today that you still have to do this kind of extraction from, and it's ridiculous. So what we really need to do is massively expand the semantic publishing and allow this data to flow directly into our databases in a proper way. And then really the other lesson here is that this task force and really this whole CETAF working group is the human connections and then this shared research questions. It's amazing the way that people have been volunteering their time. The data just starts flowing. We found out about efforts that we didn't know existed. And so really, yeah, we're talking about this opportunity. Danilo was talking about the opportunity in a crisis, right? So, I mean, we really need to dramatically rethink the way we do science in the digital age. And I think that the lockdown is showing us that. And so, yeah, just to close, Donat had this great quote of, the future is not the past. We can't waste resources to rediscover biodiversity a second time. I think that's really true. We need to kind of get our act together as a biodiversity community. And with that, I'd like to thank all of our collaborators and partners and all the task force members. Thanks.
Thanks a lot, Nathan. I really think that I fully agree that we need to do science differently, that semantic publishing will help us a lot to liberating from the scratch the biodiversity data that needs to be openly accessible for all the scientific community. So certainly we need to put a lot of efforts and also resources to sustain and support that kind of doing science for the future. Thanks a lot, Nathan. Any questions to his presentation, Deborah?
Sure, there are, the one I would start with, has there been any pushback from publishers when it comes to putting book sections or entire books online for free?
Donat is shaking his head, but he would be the better one to answer that question.
Donat, can we make his mic open?
So essentially, we don't make books accessible, we make parts of books accessible, and these parts are considered data. So over the last 10 years, we never had anybody complaining. So we have 350,000 of these treatments out, and there was no complaint so far. But it's actually, it's more, because it's, even if we take a book out and make it accessible, it's the book per se is not open, but we add metadata like this custom metadata. We add links to figures which nobody knows are inside, and to make the figures accessible because figures are open. We make links to treatments, and so it's essentially also, if you want, PR for the books.
Thanks, Donat. Do we have time for one more?
Yes, a quick one.
So Nate, or I guess also to everybody in the group, how would you, how do we change community standard of practice for like semantic publishing from the start? Where do we begin that?
Well, interestingly, I would say that, you know, this task force has been an outstanding start, right? So, I mean, part of it is just realizing, or helping people realize that it's necessary, right? And so if we can't realize that it's necessary within our own museum community, then we don't have much hope. But I think we're making progress in that front. And then it's sort of, you know, how do we communicate what we now know, which is, you know, a lot of people outside of the museum community assume that we already figured these problems out, right? You know, they think that we know everything about all species, you know, I mean,
sure.
Maybe they think there's some viruses that are unknown, but they think we at least have like the mammals down, which, you know, we certainly don't. And so like getting that message out more broadly is going to be important.
So I guess I would just finish, or the final thought to confirm with you. An example there would be, did you know about Plausi or Globi before you became part of this task force? How much did it change your perception of what was possible?
Yeah, so I didn't know about Plausi until I met Donat. I had just met Jorit right before the task force started, and then I brought him into the group actually. So that you know, it's been very fortuitous.
So that's already a big achievement. Connect communities of action.
Yeah.
That makes a lot that is a lot of the effort that we have to do, raising awareness, supporting dissemination of what it is out there, and connecting communities. That makes a lot. So yeah, thanks a lot, Nathan. We need to move forward towards the fourth activity. And that activity deals with metadata registering practices. A key element for the research into understanding the origin of the SARS-CoV-2 focuses on molecular sequences of animal specimens. So the group, the fourth group, tackled this issue, and it will be Jerry Lampher, the one participant, presenting how much they have achieved during the last 3 months. Jerry Lampher, he is the Chief Technical Officer of Elixir. Elixir unites Europe's leading life science organizations in managing and safeguarding the increasing volume of life science data being generated by publicly funded research. Jerry joined Elixir just 3 years ago, but it looks like he has been there forever after a long career with the pharmaceutical company Pfizer. He's a biologist by training, and he's particularly interested in how we can bring the power of the molecular disciplines to understand biodiversity at large. So I think we need to really be pleased to having Jerry participating in this task force. And now is your time, Jerry. The floor is yours for presenting the achievements done in this group. Thanks, Jerry.
Many thanks, Anna, and hello everyone. So yeah, I'm just going to cover the final piece of work that we've done around the molecular data and how that relates to joining up the viral sequences and natural history collection specimens. I should say, of course, that I'm talking on behalf of the group whose names are listed below. We've got a diverse set of people there. We've got mammalogists, we've got museum informaticians, we've got bioinformaticians, we've got some botanists. So it's a real wide sort of discipline. I think that was really important in terms of the work that we've managed to do. So if we think about this in terms of what was our objective, so it really fits into a much broader vision of creating a more integrated system of biodiversity and molecular data that tries to integrate multiple communities, so natural history collections, molecular biology and field ecology, that will ultimately allow much more sophisticated and cross-discipline queries. For this particular work stream, we've been thinking about how we connect natural history collection specimens and molecular data more effectively, because we believe that this will be a key method to research the origins of COVID-19 and of course also predict future outbreaks. But I think the key thing is and I was reflecting on one of the earlier speakers, that in a crisis comes opportunity. I think there's an opportunity to build on that much broader vision through what we can do trying to address the COVID-19 crisis that we face at the moment. So we've heard a little bit about this. There's the surveys going on to understand the types of questions that people want to ask. But certainly we believe that as this data becomes better integrated, we should be able to help address some of these questions. So things like the site of origin and process of viral transmission to humans, how we can plan and mitigate for future transmission, search for similar viruses in other wild hosts, perhaps predict future pandemics, discover previously unknown host pathogen relationships that may be important. And more generally, such a system we believe would be widely applicable in the fields of biodiversity measurement and conservation. If you think about specifically in molecular terms, what types of experiments might this be important for? So what types of experiments from a molecular context would you run in order to be able to inform those types of questions? Well, perhaps these are the types of experiments you might run. So survey bat museum specimens, maybe one or many species, for the presence of coronavirus sequence signatures. So look to see if you can find coronaviruses in some of those bat museum specimens. Following on for that, you may wish to isolate and sequence a full-length genome of a coronavirus from a species of bat. Maybe not from a museum specimen, but perhaps from a freshly wild-caught specimen. You may want to, based on that, you may want to screen captive bats for a particular species for the presence of a coronavirus, perhaps to understand risk of transmission. Not then thinking beyond the virus itself, there may be work to characterize the genome or the microbiome of a host species, perhaps to sequence the ACE2 receptor for the virus in several species and understand how that relates to the particular virus and how it relates to the human situation. And again, and then more broadly, develop a more integrated and broader understanding and visualization of host pathogen relationships. So this is what we hope to address. So this broader scientific questions help those be answered, and then enable these types of experiments through better management of the data, essentially. And so you may ask, why is capturing this metadata important? I'm not actually going to dwell too much on this slide. I'm just going to really pick out the top bullet, which is, without the host vouchers and the metadata, in the future, researchers just simply cannot reproduce or validate those types of experiments. So if we look at this little panel on the bottom right here, so the museum specimen is sampled, the sample is sent for DNA sequencing, the sequence generated is deposited in the repository in GenBank or in ENA. And if the voucher isn't correctly captured, the link to the voucher isn't correctly captured, you can't then navigate back. It may be hard for a human to do, perhaps it will be possible, but it may be impossible for a machine to do. And if you want to look much more holistically across these data sets, comparing between different experiments and done at different times on different populations and different environments, you simply can't do it if you don't have that metadata captured in the right way. So what have we done? So Pam did share this earlier on her talk at the beginning of this session. So with 3 phases to our work. So we thought about the high-level metadata requirements and definition refinement. So what metadata do we need to capture? We thought about what guidance and tools can we generate in order to help the community capture those metadata in association with their sequences in GenBank in a consistent and reliable way. And then, as we're just starting today, what actions do we need to make sure that we socialize this across the relevant scientific communities? It's all very well having recommendation and guidance, but if it's not adopted, then we haven't been successful. Okay, so let's think about the first 2 of those outputs that we've been thinking about. So the metadata. What metadata do you need in this instance for a viral sequence associated with a natural history collection specimen? So we had, we thought about this for a number of weeks. We sought input from quite a wide range of people, including people outside of our immediate work group. And we came up with a long list of metadata that would be great to capture in these sequence repositories. However, this sort of generated a bit of a light bulb moment for us, which is we realized that all of this data is actually captured with the host, with the voucher in the museum specimen, or at least it should be. And of course, this also relates to the second work stream that we heard about in terms of capturing the metadata associated with the museum specimen. And so what we concluded was that ideally in the future, what will happen is that we won't have to recapture all of this data in, let's say, GenBank, that we would be able to access that data via the link from GenBank to the museum voucher specimen and the associated data that holds. And so in the end, and we've settled on a number of key metadata that we'd like to see captured, including host geolocation, host voucher specimen ID, and of course the host species, as we believe those are important if you want to data mine and navigate through from a molecular sequence perspective rather than from the museum voucher perspective. I should say that we weren't starting from a blank sheet. Many of these metadata that we identified as important already were available within GenBank and could be and indeed were used by users, by the community in this field. However, our observations were that there were some key fields that were missing, and in particular host voucher specimen ID and the host geolocation that particularly relates to viral sequences of this type. But also that the data fields were not used consistently. So some publishers would, some data publishers would use much of the data, or they'd use it, or some wouldn't, and then sometimes it was used inconsistently. And of course, what we need for the data set to be truly valuable is for it to be consistently applied and consistently used, because then the data becomes ultimately reusable. So if we then go on to think about, if we've identified what metadata we'd like to capture, how can we facilitate the community to do this as easily as possible? And we're doing that through generating a GenBank template to aid with data entry. So this would include our recommended key metadata, including the new data fields that we've added. I should say this is by no means final. There remains, this is work in progress. So this is not available today, but we hope that it'll be available in the near future. Our colleagues at NCBI are working on this as we speak. I've indicated there just a sample of the template. Many fields are hidden for the sake of clarity of the presentation. You can see that from the column headings that many are missing. But just to give you a flavor that you can imagine, if you generated a lot of sequences from, let's say, museum specimens using a consistent experimental approach, it'd be easy to complete a template with all the relevant metadata, such as the bat species, the host species, the host geolocation, the host specimen voucher, including that can now be, or will be able to be, a link, a URI, rather than just only flat text as it has been in the past. Of course, links to the viral voucher specimen as well, and of course your sequence data. This would be relatively, we hope, straightforward to complete, and it would aid with consistency across experiments. Of course, this is an ongoing process. It will be improved or updated over time, but we would hope that this will be the basis of something that's really usable by the community. So what are our next steps? Well, clearly we need to finalize and refine the template to make sure it's fully usable. We have not tested it as yet, it's work in progress. We need to really think about how we market and communicate this, make sure that our recommendations are adopted. So we're thinking about dedicated webinars later in the year, conference workshops. Clearly not the ideal time to do that, but it's an avenue we'll need to look at over the months and so on ahead. And of course, we think about a publication in a relevant journal. And also, of course, we think about extending it to non-COVID related fields, because much of the learning that we've got here would be applicable elsewhere. So perhaps epidemiologists who often deposit vouchers could think of this in the same way, use this type of approach in the same way. And then extend it more widely to the natural history community to integrate, perhaps could even consider integrating it into the loans process, so that if a museum loans process was put in place that involved generation of sequence data, it would come with the agreement that that sequence data would adopt a certain model of metadata capture. So finally, so what are the key messages? So in the short term, we're trying to improve and standardize the metadata that's captured in association with coronavirus sequences derived from natural history collection specimens, e.g. bats. In the longer term, we would hope to extend these learnings more broadly across the intersecting communities of natural history collections, molecular biology and field ecology, as we think this is of fundamental importance to making sure that we generate the impact of these collections and the molecular biology data that's generated from them. And of course, finally, as this is work in process, we plan future webinars to review this in much more detail later in the summer or the autumn. And I'll stop there.
Thanks a lot, Jerry. This is really important to see how we need to really include practices, good practices for metadata registering and improve and enhance the information related to the species. So thanks a lot. Any question to Jerry, Deborah?
Yes. And you can see in the chat as well, John Yin asks, over the last decade, it's become increasingly clear that virus-host interactions are not limited to two biological entities, virus and host, but often mediated by other species, resident bacteria, viruses of the bacteria, defective interfering virus genomes, and particles. To what extent are specimen collections seeking to account for broader host-associated microbiomes and viromes?
So I think that's a question for the second work stream. I'm not seeing Kendra on the, don't know whether she's still online.
Well, you know that even though some questions might be tabled today, if we don't have time or the person is not here so far, we will, as I said at the very beginning, we will have the time to come up with the due responses, and we will use our social media to channel those responses and disseminate it. So please don't hesitate to still place your questions, your remarks in that Google Doc, and we will come back to you at any point, because I think we are running out of time. Well, basically we are already 12 minutes over the scheduled time. So if there is any other burning topic to raise to Jerry, Deborah, if not, we shall conclude.
Kendra is here. We don't have time to respond to that?
Here, I can respond to it. This is Joe.
Well, Kendra is here too. Sorry, Joe.
Yeah,
and I think people
Joe, yeah.
can leave if they need to, and then we can just say, come back to the recording if they, and that way we're respecting their time, but others can stay if they can.
Okay, well.
Joe, feel free to jump in.
I think this is the point that's been emphasized repeatedly throughout the a couple of the presentations, and that is that museums, if they begin to build holistic collections, will become central to the science that is much more integrative, that allows us to look at not only a virus in a host, but also the microbiome, the other microbiome elements in that host at the same time. And it's a very different way of doing virology than what's been done in the past, where no host is preserved, and so you couldn't actually get at the question of the rest of the microbiome. And so again, this is clearly the future for collections in their ability to integrate across many different angles or many different perspectives in issues like this.
Thank you for that. There is another question in here for standardizing the metadata, Jerry, so that it can be shared across collections. Is working with TADWIG, the Biodiversity Information Standards Group, part of the next steps?
Yeah, so I think it probably could be. I might ask Deborah whether you'd like to address that, or Pam.
I just say yes to that, so we can keep it short, and you can contact me. Yeah, sure.
Okay, well, I think we need to stick to the structure that we envisage, and I think we have a lot to say. I'm sure there are a lot of questions that may rise today or may come to your heads later on. So please feel free to contact us. Please go ahead with your remarks, your questions, as I said, your comments to either one of the presentations or the overall initiative, and we will try to give you the response. I think we are heading to closing this event, and I really think that this is now the time to perhaps give the final words in, and I wouldn't like to close this event without thanking several people that I would like to mention. First of all, the CETAF and the DiSSCo teams, especially Celine Casarino, our communication leader, for disseminating, for all her efforts in disseminating and outreaching this task force from its very beginning. And of course, thanks Deborah for all your big support today and making this event running smoothly and so effectively. I would like to, of course, address every one of the participants in the groups, but I cannot name one by one. So I really need to start thanking especially Pam, our keynote speaker, our 4 presenters, Danilo, Kendra, Jerry, and Nathan, but this is extended to the entire group of experts that have been on volunteer basis, dedicating so much of your time to doing the work under this task force. Thanks a lot for your passion, your motivation, your enthusiasm every single day that we have met and that we expect to keep meeting in the near future. So I would like also to thank all our supporters and collaborators, starting with Elixir, our colleague in the European Strategy Forum as another research infrastructure together with DiSSCo. I would like to thank GBIF, Spinach, TEDWIG, IDIC Bio, Globi, the American Society of Mammalogists, and of course, Platzi and Pensoft for all your support, for your total engagement, and for helping us in making this task force a reality. I would like also to thank Dimitris Koureas and Wouter Adink from DiSSCo and Naturalist Biodiversity Center in the Netherlands. They are my teammates in organizing this task force, and I really do appreciate their hard work in making this adventure not only a reality, but also a pleasure. To everyone, additionally, last but not least, to all of you that have participated in this event. This is not a goodbye, it's only a way to say see you later, because I would like to encourage to participate in the task force. Join any of the activities that you have heard about. Please keep tuned. because we will follow up on all your comments. And please go to the social media of CETAF, of DiSSCo, of all our collaborators, because you will see this event and all the videos of people that has contributed to make, to disseminate to widely our task force, and also the way we are going to continue with our work. So really, I would like to finalize this event with a video. We have posted a video. This is a video from DiSSCo linked project iStick that encapsulates the real message that we would like to finalize with. The importance of the natural science collections in defining, in making science excellent, and of course being the pillar, the robust and critical evidence for many other disciplines that need to build on top of our data and our information. So this video, I think the link is posted. Celine, is it?
Yes, the link is on the chat, so you can have a look, watch it.
Either look at it now.
We will just now share it, but you have the link if you want to keep it, watch it again and again.
That's right. So yeah. Thanks to you all.
She's sharing and supporting me to share. Just wait 1 second.
Okay, thanks you all for your commitment, for your participation, and have a lovely afternoon. See you all soon, I hope.
The European Natural Science Collections are a unique source of information about life on Earth. These collections contain vital information to understand current challenges, such as biodiversity loss. They provide evidence for implementing policies like the European Green Deal and help model the impact of climate change on our planet. Europe is a global leader in collections-based research, hosting 80% of the known bio and geodiversity, preserved in approximately 1.5 billion specimens, and a large committed community including over 5,000 scientists working on them. To study these collections, every year over 25,000 people travel, while more than 800,000 specimens are shipped from one country to another. This current inefficient system comes at an annual cost of 70 million euros and causes a significant ecological footprint. DiSSCo enables the transformation from this fragmented landscape of the natural science collections into an integrated knowledge base that provides urgently needed and interconnected evidence of the natural world.
DiSSCo is to bring together the natural history collections virtually, so that any user of the data that is contained within the natural history collections can just use a one-stop shop.
Specimens are containers of multiple types of information, including genomic, biochemical, geographical, ecological, and everything in between. With DiSSCo, physical specimens and their data transform into a new concept of digital objects. They become digital specimens. These digital specimens will contain all the information extracted from their physical counterparts. The data will become widely accessible and fully interconnected to allow scientists to contribute to the understanding of our world at an unprecedented scale.
DiSSCo is a vision of an operating research infrastructure, but we are not there yet. We need to think about how to do various things technically and how to adjust our modes of operation. We had a design study for DiSSCo iStick, which stands for Innovation and Consolidation. Natural history collections contain a vast array of different kinds of objects. We have anything from microscopic slides to big mammals, the extreme being the blue whale. So we have numerous different tasks of developing a workflow and a methodology to capture both images and data from those specimens. It's very important that digital data resources are made fair, that is findable, accessible, interoperable, and reusable.
DiSSCo will operate under an innovative business model to underpin a large-scale initiative that creates one system to prioritize what to digitize, where and when, one shared strategy on collections development, and ultimately one European collection of scientific assets. As the entry point to this European collection, DiSSCo enables scientific discoveries that will also benefit other scientific disciplines, industry, like agricultural petrochemistry, and society alike. To provide these services, the DiSSCo research infrastructure plans to be fully operational by 2026. DiSSCo, providing robust evidence on our planet's natural diversity.
Hope to see you all soon. Bye.
Thank you all for coming.
Thanks very much. Bye-bye.
Bye.
Bye.
Thank you.
Bye.
Thank you.
Bye.
Goodbye.
Bravo.
Bye you all.
Je vais dire merci à
So let's leave, and we will make a wrap up in our daily Zoom meeting, okay? Just 1 second. Bye.
Great, bye.